Thank you. Nonetheless, this is an important discussion. It's a pleasure to be here, and I appreciate it. And while for a while I thought about talking about this year's best fishing lures, I thought that it probably would be good to stay on topic. And I see that my plan to short out all the clickers worked quite nicely. No clickers today. Oh yeah, thanks. Well, thank you. So I probably don't have 7 minutes, but I have a few things that I'd like to share with you that can stimulate some thought and provoke some discussion. So if you think about some of the limitations, and of course no one could deny that epidemiology and foodborne outbreak investigations has brought us a long way and has been a driving engine for solving many. But if I could point out some of the caveats associated with shoe leather epidemiology, you might want to point to the fact that we still don't really have a mechanism in place that solves all foodborne illness. About 1 in 3, some estimates maybe even a bit higher, of foodborne illness do remain unsolved, where we either don't find an isolate or a vehicle that caused it. Some of that is due to the fact that by the time we get through case reports and interviews and we work through back to the actual actions of tracing back to source, we are often on the back end of the curve, back here, instead of up here where we'd like to be. So by the time we're back here, if you're trying to track down a tomato event, there are no more tomatoes left. So they're all gone. So it's very difficult, and that's something that I think that new technologies could help augment us more towards the front of that curve. Another issue would be I think this perhaps could be the most important issue, is the accurate recollection of events in the human brain. And that's why you'll see I dappled this slide with a few cartoons about the difficulty that humans have in recall. And particularly once you get beyond 3 days, most of your short-term memory has emptied out. We had a foodborne event in 2003 in Washington, D.C., and it was actually at an event that FDA was part of, and many of us got sick. And they called me, the Washington Department of Health called me and said, we're trying to figure this out, so we'd like to interview you. Did you have the soda with no ice? And I said, actually, that's correct. And they said, well, what about the soda with ice? And I said, yes, I had that too. And then they said, well, what about the Sprout sandwich? And I said, yes, it was delicious. And they said, well, what about the ham and cheese, the cold cuts? And I went, also very delicious. And I said, the potato chips probably didn't do it, but I can tell you the 3 kinds I had as well. So in the end, they hung up on me. They actually said, you're absolutely no help at all. So I wished them luck. But in the end, I was amazed that, for me, recollection was easy because I ate it all. But for some people, it's not so straightforward. And so that's an issue. And then the issue of in-product testing is always difficult because we know, Peter Gerner and I were just discussing this, we don't have the ability to do effective in-product testing here in the United States. There's way too much product. And so it is sporadic. And sometimes we have good fortune and we hit the right chord and we find it. Sometimes we don't. But it's difficult in relying on in-product testing for root cause. The other thing is that classical approaches tend to require a critical mass. You have to know that there's something wrong, and it usually requires a certain trend to see that something's wrong. And so I consider that to be perhaps a caveat as well. Here's one that my colleague Errol Strain provided me from FDA. We were chatting about it, and he said, "It is quite difficult, you know, to standardize questionnaires across international boundaries, making it very difficult to compare data within the United States to with outside the United States," which was a good point as well. And many times traditional shoe leather approaches require you to lay down a working hypothesis of the geographic distribution or the proximity of people to a certain event, which would make sense. I mean, clearly, taking the handle off the pump in the original Epi case of John Snow required that everybody was in a certain radius of the pump itself. And then lastly, it gets difficult to go beyond a certain period in time back, maybe 3 to 6 months. Even with certain conventional subtyping tools, you can go back 3 to 6 months reliably, I think. And so these are the caveats that I wanted to bring forward to discuss. Now, if in the new age of microbial forensics and molecular epidemiology, or what we call genomic epidemiology, we have a method that raises the degree of certainty and the degree of confidence, one could argue to the point of the asymptote of truth. And so that sort of confidence is something that we believe will catapult genomic epidemiology into the forefront in its use from this point forward, but it's not novel by any means. The Florida event in 1990, where the dentist infected his patients with HIV, was worked out first by whole genome, or not whole genome sequencing, but genome sequencing. The Amerithrax event with the postal letters of 2001 was also elucidated by whole genome sequencing. That was back when we had to do it the hard way and it cost a lot of money. There were many hospital-acquired infections that have been traced back over the last decade, particularly in the realm of MRSA and VRSA, using whole genome sequencing. And the Haitian cholera outbreak also. Whole genome sequencing did show a rather unexpected match between the Haitian illnesses and the Nepalese peacekeepers who came over from Southeast Asia. So in many ways, it has already shown its value before it even got into food safety. To think of whole genome sequencing is to think of it more like the Game of Thrones pedigree turned on its side. It's just a family tree. A phylogeny is a family tree, just showing who begot who and who's related to who. And with it, you can start to discern very quickly who the players were in an outbreak and what the potential sources were behind it. The FBI does this. They do this all the time. They do this in criminal cases. They actually don't even use all the data like we do. The FBI uses 13 sites. The FBI CODIS database can use 13 markers in the human genome and actually go back and type almost in every case the origins of the individuals that they're typing. 2 minutes, 1 minute? Is it 10? 0 minutes? All right. We're looking at 3.5 million sites. That's a lot different. And then lastly, just my last slide is to show you the examples. We sorted through all the ingredients already. We've shown you can sort out multi-ingredients. We linked up an outbreak halfway across the world. We've probed back in time 3 times and brought cases in linked that we had no idea were part of it. We sourced down to the farm where the eggs were being laid, and most recently we bypassed the food vehicle in its entirety and still made the case and did the regulatory action. So it's an argument to be discussed, yes? Thank you.
Dr. Weidman, is shoe leather epidemiology dead in the age of whole genome sequencing?
It's not. But I feel like Germany playing Iceland in a soccer game where everyone expects Germany to win 15-0 based on the pre-vote, so it's not that easy an argument to make because they're going to win over the remaining 5 people. So what is the issue? Why do we still need epidemiology? We can have identical bacteria that match 100% over their whole genome in many different places. I came in from my Cornell dairy plant. I have listeria on my shoes. It divides. Eric steps into it, goes back to wherever he's going to go. The same listeria is in this other place. Without epidemiology, we would not know that. Here are the data. Here's the theoretical background. Bacteria populations are gigantinormous, what I'm going to call identical twin issue. Bacteria divide, they're still identical after they divide. how quickly is it that bacteria do become different? There are a couple of different calculations we can do, but the idea is it probably takes between 450 and 9,000 generations. So bacteria doubling, 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 before they're even different in one single nucleotide variant. I have a citation there from a paper that was actually not food safety, but about a hospital environment, Clostridium difficile. And there is said that we had, with whole genome sequencing, we have 0.749, so less than one difference over a year for a genome. So even in a year of Clostridium difficile, multiplying in a hospital, they may or may not be different over 1 year. That is the challenge we face here. So practical observation, this is the theory. Practically, is it relevant? We published a paper with CDC almost 10 years ago, which showed that we could find the same Listeria monocytogenes, more or less, by whole genome sequencing in the same plant over 12 years. We could find Listeria differed by one single nucleotide polymorphism, stayed the same over 12 years. That just shows how stable these genomes are and how even with whole genome sequencing, we can find identical organisms. Here's a short chart of that. It showed that we have, in one plant, we had isolates from 1988, from 2000. If you look at the most similar one, this food isolate and this here, no differences. There's one difference then in that human isolate. So if the known isolate, this human isolate, this food isolate, one difference over 12 years in the same plant. We just published a paper where we did whole genome sequencing of Listeria monocytogenes from more than 10 retail delis throughout the US. We found Listeria that differed by less than 3 SNPs, less than 3 out of 4 million nucleotides in retail delis in 3 different states. I'm not talking adjacent states either. We have exactly the same Listeria monocytogenes in retail delis. So if I don't have epidemiology, how do I know which of these stores is the source of the outbreak? Or is it in the upstream chain? I wouldn't know it without epidemiology. There's precedent to this. And this is the comparison. So if you remember one thing, remember, bacterial is a gigantinormous identical twin issue. And even, Eric cited FBI, even when we look at criminal lawsuits, we cannot differentiate identical twins by DNA fingerprinting. Here are a couple examples, and this has been very common in the literature. So we have identical twins. We find DNA of one twin at a murder scene, as a scene of a crime. Which of the 2 twins is it? I cannot differentiate it by DNA evidence. Why? Because they're identical twins. These cases went to court, mistrials. Why? Because we could not place the person in the place. We didn't have epidemiology. We don't know which twin is there. We don't know which one did it. We cannot convict that twin. That's exactly why we need whole genome sequencing, and why we need epidemiology. The last example, we need whole genome sequencing first to then do epidemiology. The final exhibit is a study that really illustrates the practical issue. So this was one where Salmonella was isolated from fields in the Delmarva Peninsula, and the Salmonella from geese feces actually matched, was the most closest match to, there's some interesting dynamics happening here, we'll figure that out later. There's some finger pointing here. Yeah, that's fine. I'm being careful, come on. So we find the closest match between Salmonella from geese feces and a human outbreak. But what does that mean? Well, when we look at flyways, geese can move very, very quickly, very long distances. Does Salmonella become different over a flyway, or while a goose travels hundreds of miles? There are not enough generations to happen. So there are many, many mechanisms, hypothetical and real, how Listeria, Salmonella, with identical genome sequences can be dispersed over long distances and can be found in different places. We need epidemiology to differentiate those possibilities to have accurate and appropriate outbreak attribution. Bottom line, if it doesn't work for identical human twins, it doesn't work for bacteria. Thank you.
Dr. Brown, you have 3 minutes if you'd like to add some additional comments.
As a matter of fact, I would.
The ladies down here will tell you when your time is up.
Thank you. That last paper was a bit personal. So, on Martin's first point about the issue of walking away and having it on your feet, and that could distribute it, and that would make no sense from an epidemiological focal point, I agree. I can't rebut that. On the second point, however, of similarity remaining strong over long periods of time, in the BMC genomics paper that Mark Allard and colleagues published in 2011, the replicates were done at the plate level, the colony level, the DNA prep level, and the machine level. And the titrations of mutations were taken into account, and we now know that at least in the case of Salmonella, we can accrue about 1 to 2 base pairs per overnight culturing event in Salmonella. That means that sameness to us is 1 to 2 base pairs across the whole genome. Anything beyond 5 or 6 or 10 would get us out into the realm of completely different. And so there is going to be an argument about how to titrate what sameness is. But we do have enough sensitivity, I would argue, that you can find the differences. It's now figuring out what those differences mean. Now, Martin is discussing Listeria. I'm discussing Salmonella, so we do come in from a couple different angles. The other thing is in a plant, we looked at a plant too over 10 years, a dry factory cereal plant. We found 6 SNPs between 10 years worth of isolations from start to finish over 10 years. That's enough differences that you could sort out the old isolate from the new isolate, but similar enough that you could show it was still from the same factory even after 1 decade, the signatures were there. Not adjacent states. Martin says if they're not in adjacent states, what does that mean? How can you link it back if it's not adjacent states? I really don't have a strong scientific rebuttal on that, other than to say that there are trucks now with 18 wheels that tend to haul things from a certain point across the country beyond single states. So we shouldn't be surprised that we see things beyond single states. As far as twins, the twins argument, that's true, but I would argue that over time, the twins can diverge much more quickly in bacteria. But in people, Martin was right, and I probably would have just had to put them both in jail, just to be right back. And then lastly, while Martin is correct about many flyway geese that migrate between the northwest part of the continent and the southeast, these geese in particular that live around AREC are resident geese and can be found there year-round. Thank you.
Dr. Weedman, 3 minutes.
Okay, thank you. So I'm going to do a little bit of a rebuttal on Eric's point on why we should not do epidemiology. And the bottom line is epidemiology difficult in foodborne disease outbreaks? Yes, it is. Does that mean we shouldn't do it? No, it doesn't. It is difficult. We need money. We need resources. This is why this is an important debate to have, because we need the resources to do epidemiology and to do it right. And in all reality, with whole genome sequencing, the epidemiology tools, the resources we need for epidemiology need to actually be better because we detect more clusters, which we need to follow up on. So we need to do the difficult thing, and we need to have the resources to do that difficult thing.
I agree.
So we would like the audience, if you've got some questions for either of our speakers, we do have a few more minutes left before we will vote again. So if you do have a question, would you please step to the center aisle and use one of the microphones?
Hello, Mark Allard, FDA. I have a question for Dr. Brown, and congratulations for decimating the opponent, though you should go a little more easy on him. Are there any examples where the whole genome sequence provided evidence that then the epi missed, and that the whole genome influenced and guided that?
Well, there are examples where, because of the sensitivity, we've been able to find it through some routine examination of the ever-growing trees. And in particular, once you get beyond 3 or 6 months back, it becomes quite challenging. And one thing I'll say that I think has been powerful about the whole genome approach is that it's actually been able to bring in illnesses as far back as a year 2 years in time. And that was, I think, something that one would have to argue is very important and very powerful about the new tool, definitely. And thank you for your compliment on my debate strategies. I'm sure Martin will have a comment, but since I've got the mic here, I'll just go first real quick. So I think you actually answered what you were asking, which is very important. The supply line monitoring for industry is so absolutely going to be key for whole genome sequencing applications. If you find matches between supply lines and multiple processing facilities, you've kind of already pointed back to where you think it might have come from. You've got a common supply line allele, a common genome in the supply line. That would be your most parsimonious argument. If enough time has gone by, I believe that you will be able to differentiate resident populations across different processing facilities. But as Martin pointed out, and I agree, if it's a recent introduction, you won't be able to for a while. So that is a caveat.
So there's a specific challenge in addition to this here, is that we don't really know how often bacteria multiply in different environments. So on two extremes, if I have salmonella, live in a dry plant, it might only double 100 times a year. On the other hand, if I have salmonella living in a poultry breeder flock or in a feedlot, that salmonella might multiply 10, 20,000 times over a year as it rapidly infects animals, multiplies. So the number of differences can be hugely different over time because we don't have defined generation times like we do in humans, for example, where we can calibrate by that. We cannot do that. So I think the differentiation is very challenging. I think, as Eric said and as you pointed out, most likely we will have an issue upstream in the supply chain, and we need good epidemiology to say, okay, if we find the same or very similar whole genome sequence in different places, what's the commonality in the supply chain, and where is the actual source of these? Or sometimes are these simply common whole genome sequence that could be found in very specific regions, which also does happen.
Thank you. In the effort of time, we'll take one more question for either of our speakers. So we have two folks standing waiting.
I'll go, I guess. Hi, Christina Ferrer, FDA. So this is more so a question to both of you. It seems that obviously one can't exist without the other, but for so long the focus has been on the epi portion, you know, the boots on the ground. Do you think that the roles have reversed now and that the whole genome sequencing should come before the epi instead of how it has traditionally been, where it's been, you know, the outbreak investigations on the ground, then confirmed by the whole genome sequencing that supports the you know, hypothesis of the epidemiologists?
You're making a very, very good point, and I think the paradigm is shifting how the two interact, whether it goes exactly the opposite way, or ultimately they truly have to work hand in hand, I think. The problem is if whole genome sequencing comes first, if recall comes first, and then we do the epi, we do run into issues with recall bias. You know? Once we have, and we see this, and it's one of my concerns, if we have a big press release saying XYZ product is recalled, and then you do the epi afterwards, then people will remember that they ate that food, even though they may or may not have. So I think it needs to go in parallel, and we need to revisit how these two disciplines work together more effectively in order to take advantage of these tools.
Yeah, and just to add one sentence, just to say, yes, it is a new tool. It brings a new capacity to it. To deny it would be wrong as well. Sometimes it may provide leads that merit follow-up investigation, and I think that's important.